No? Okay, now you hear me. Fantastic. Well, thank you for the opportunity to present. And I'd also like to say a very big thank you to the CCP-EM community for advancing EM and for generously sharing this with the community. Many of us have built our biological insights on the tools that you are developing. So thank you. In my lab, we're fascinated to understand one of the free-coded polymers of life, messenger RNA. Messenger RNA carries our genetic information from DNA to protein. And it is important to understand how messenger RNA is produced and how it wanders through its life cycle. We do this in our lab by combining structural biology tools with various other methodologies. And today, I'd like to present to you just one part of the lab where we focus on a question that 4 wonderful people have contributed to. Belen, a former PhD student, Matthias, a current postdoc, Ulrich, who just left to start his own lab, and Max, also a current PhD student. And so together, they have tackled a really interesting problem in gene expression in the human nucleus. And so the question is, oh, apologies for the nuclear pork complex fading out. The question is, how does messenger RNA exit the nucleus? And a particularly interesting feature of that question is, how do you achieve this process to be both permissive to the fact that messenger RNA in the human cell is produced from 20,000 different genes and varies in sequence, in length, and in processing history? And at the same time, how can this process be selective for only the mature mRNA species and not immature precursors or any other type of RNA, where you could imagine that if exported.
All right, everybody. I think most of us are back here. Apologies for that. We're going to continue the session, and well, the talks are hopefully going to be the same length, so I hope you welcome back Clemens Plaschka with his talk.
Okay, I'm extremely excited to continue my talk. So where was I? I was trying to excite you that understanding this question of how messenger RNA is identified selectively and yet permissively to allow the selection of all mRNAs produced in the nucleus to get out for the purpose of translation. And so how does that work at the molecular level? That is the question that really guided the first few years of that part of our lab. And so what had we known was that from 30 years of biochemistry and genetics, that there is a set of factors that are conserved from yeast to humans that are necessary for the export of messenger RNA. However, the precise molecular activities of these factors, whether they are part of the same pathway or whether they are part of parallel distinct pathways, and what exactly they do, that had not been clear. And so we wanted to take a structural biology guided approach to try and add a little bit of insight into how these complexes and components might work and what they do in the pathway. And so we began with the first question, which is, how is a messenger RNA protein complex recognized in the first place? And so the messenger RNA, during its biogenesis, it is capped, spliced, and polyadenylated. And the successful completion of each of these steps leads to the binding of a unique set of proteins that mark the completion of these individual steps. And so while this is not sufficient for export, it is presumably the substrate for the pathway. We had known from previous work that this 3-component system, and I keep the color code distinct, AliRef or Ali for short, the TOE complex, and UAP56 or UAP for short, were known to be components important in the sensing of what a messenger RNP is that should be exported, but how is not clear. And so we took an unbiased approach. We, in this case, from about 20 liters of mammalian cell culture, purified at steady state complexes engaged with this packaging machinery or recognition machinery with the steady state pool of expressed mRNAs. And while I skip the RNA sequencing, what we could essentially see is that we capture what we would expect to be typical mRNPs, identified by various proteins in black and orange and so on, as well as the full packaging / recognition machinery in colors of green and so on. So to understand how these components might recognize the mRNP, we purified most of these proteins, around 40, and carried out systematic interaction studies in vitro in the dependence of RNA and ATP to ask what binds what. And from these studies, we identified one interaction that excited us quite a bit. One of these key factors of the pathway, aliref, engaged with this trimeric complex only when this complex was engaged with RNA. Now, this trimeric complex forms an assembly that is called the exon junction complex. It is deposited by the spliceosome after every splicing round, making it one of the most frequent markers of a mature messenger RNA. In addition, we noticed that in vitro, this seeming one-to-one stoichiometric assembly formed various means of oligomers. And so to understand how this one component might recognize a feature of the mRNP and mediate multimerization, we solved its single particle structure. In this case of a hexameric assembly, where you can see here in shades of orange, the exon junction complex bound to a short piece of RNA. And in purple, you can see this protein aliref. And to us, what was really fascinating to see is that a single aliref protein, and now I will not show you the nitty-gritty details, has the ability to simultaneously bind 3 separate exon junction complexes. The structure therefore helped us explain not only how recognition might occur, but that recognition of a feature, a very frequent feature of the mRNP, might also coincide with compaction, with bringing neighboring exon junction complexes into proximity, perhaps to facilitate the packaging of the RNP. And biochemical and cellular assays confirm the importance of these interfaces. Now, that was great. That gave us a first step towards understanding recognition of a key feature of the mRNP. But the question is, how do you go from this assembly towards the full mRNP? So while recognition, and we by now have even more data on this, is mediated primarily by this protein, we also know that there are additional components that are absolutely necessary for recognizing an mRNP for export. And that's this 2 megadalton assembly combined of this TOE complex and UAP6. How do they engage with mRNPs? What do they do? And what does an mRNP even look like in 3 dimensions? So to address these questions, we then proceeded, instead of with the reconstitution system, which we could build bigger and bigger, to native mRNPs. And at this step of processing. And we asked, what do they look like? And so this is in vitro tomography. So this is the purified sample studied by tomography. And so in this movie, you can see a variety of particles that are monodispersed. And each particle we think is likely to be a single mRNA with proteins attached. We can analyze these data using well-established pipelines to gain insights into what they are composed of. And so I'd like to focus your eye on this one particle. In the center, you see this diffuse density, and this is the mRNP, visualizing for us with under native cryo conditions, what an mRNP can look like. They form these oval-shaped globules. And on the surface, in this case in green, is marked the TOE complex, one of the members of the packaging complex. Now, to put this only into a relation to the nuclear pore complex here in the dilated form, we were interested to see that at least the average particle that we captured would be in principle compatible with the diameter, of course, you must imagine, filled with FG repeats of a dilated nuclear pore complex. If we look more closely, combining a single particle structure from a reconstituted assembly as well as the native assembly, we can visualize how this packaging machinery binds on the surface of these mRNPs, presumably to recognize them for the next step. It also suggests conceptually to us that mRNPs might be organized in a layered fashion. Here now with the first example of such a layered organization by the TOE complex itself. So what is this good for? So far, I focused on a very quick run through of how the first 3 components of this pathway can bind and recognize the mRNP. We have much data that I did not show you that suggests of how compaction is facilitated by aliref as well as recognition. This then forms a multivalent assembly where the allograph protein can bring in the next members of the pathway to form this Tau-UAP mRNP complex. And now the big question for us was, what happens next? The only thing we knew was that this assembly cannot export the nucleus. What must happen is that the green complex has to dissociate. We know that subsequently, the mRNP must somehow dock at the nuclear pore complex. Again, not in view, but it's there. And finally, once the mRNP has docked at the nuclear pore complex, it must also be released again into the channel such that transport can occur. And these are the questions that we tackled next. The first question, once assembled and recognized, how does this assembly disassemble? And so to generate a hypothesis on this, we looked very closely at the structure of this part of the native Tau-UAP mRNP complexes by single particle analysis. And so here you can see in green this 2 megadalton assembly that I mentioned that binds on the surface of the mRNPs. And while you do not see it from this view, this is in fact a tetrameric assembly. And each of these ends here, the contact site to the mRNP is repeated 4 times, presumably adding avidity and thereby specificity towards what an mRNP is. Now, to understand this question of how would this green complex Tau release from the mRNP and why did it bind in the first place, we looked more closely at one of these 4 binding sites or contact sites between the packaging machinery and the mRNP. And so this is a zoom in. And so what you can see is that the protein allograph, and this is shown for completeness, is what would contact features of the mRNP. This generates a high concentration of these short peptides that in a second step can bind to UAP56. That's this enzyme in the middle. And finally, there is another contact here with a subunit of the Tau complex. And the reason why we think this is important is because Tau not only multimerizes, meaning multiplies this binding site times 4, but it also positions these lobes for optimal engagement with RNA. And so let's talk about that for a minute. We knew that this assembly has to disassemble, but how would it do this? What is really cool is that this enzyme, UAP56, is a member of the so-called DXD box ATPases. It can exist in 2 states, an unclamped state, and in the presence of RNA and ATP, a clamped state. And so you can think of this as a molecular switch. And so because in our structure, we observe the unclamped state, we hypothesized that perhaps it is this transition towards engaging RNA that would lead to remodeling of the complex. And this we've tested in various assays, and I'll show you only one, where we used a surface plasmon resonance-like method to measure binding affinities. We mobilized the recombinant Tau complex on a chip and wash in UAP56, and we get binding kinetics of reasonable affinity. Yet if we pre-incubate this enzyme with RNA and ATP, the affinity is non-detectable, providing the first suggestion that indeed the engagement of RNA might be what drives remodeling of this recognized assembly. While I will skip the cellular, genetic, and biochemical data of both reconstituted and native assemblies, what we could show is that the addition of ATP and another factor in the pathway, whose role was not clear to this point, are necessary to efficiently disassemble this Tau complex. And at the same time, we were surprised to identify another previously not known intermediate in the pathway. And in this new intermediate, what has changed is that this enzyme, and I should mention, this is the most important enzyme in terms of conservation and essentiality in the pathway, from yeast to humans, goes from this unclamped state to an RNA clamped state. And so we reasoned that perhaps a subsequent step of the pathway would be sensitive to the clamping state of UAP6 as a checkpoint that might have been passed in this assembly. So to identify putative interactors of this clamped state, we carried out a binary AlphaFold screen. So we took all proteins enriched in a native IP of this protein UAP, and then did a binary interaction screen with which we were happy to recover interactors that we had characterized, paralogs that are very diverse of the allele protein. These paralogs, we hypothesize, give promiscuity to the system to recognize perhaps substrates that are different from the regular substrate. And then we were really happy to see that among the top predicted interactors are these two proteins colored in blue. And these got us really excited because these two proteins are part of another complex that's called TREX2. For reference, this is a nuclear pore complex. And the TREX2 complex in human cells is constitutively associated with the nuclear pore. It had been known that it is important for mRNA export, but exactly what its function is in the pathway was not clear. And so the prediction by AlphaFold suggests that perhaps it is this clamped state of U56 that is read out at the nuclear pore complex to allow the docking of the mRNP. And I will again skip the cellular and genetic data, but I show you just the structure of a reconstituted assembly of a key feature of the TREX2 complex. That's this V shape in blue. In this case, captured, engaged with a part of U56. And there were two features of that structure that really excited us. The first one was at the very end terminus of this protein is a very unique, but in evolution almost invariant sequence that binds to TREX2 and is necessary in vitro for an appreciable amount of the affinity between these complexes. And we had known was important for export, but has no known role in the earlier steps. The second feature of interest that we found was this loop here. And so for this, I zoom out for a second. When we reconstituted this assembly, we assembled the RNA clamped state. And so our expectation was that minimally what we should see is this, both of the lobes of UP engaged on the RNA substrate. Yet what we saw in the structure was this, suggesting, and by structure superposition, it is clear that this loop in TREX2 actually wedges in between the two Reg A lobes of UP56. Now, if that were correct, that would be rather exciting because it would suggest how at the nuclear pore complex, this TREX2 component may not only be the site of docking for the mRNP to the pore, but also the site where U56 would be released. And this would be important because it would release this tethering point and allow entry of the mRNP into the channel. So if that were correct, you could make many simple biochemical predictions. One being that TREX2 should stimulate the ATPase activity or at least turnover of U56. And so this is the only biochemical assay on this that I will show you. And so what you can see is that U56 is an extremely lousy ATPase. And that is because the clamp state is very long-lived. TREX2 is not an ATPase. If you add TREX2, ATPase turnover increases. And if you introduce a single point mutant in this loop of TREX2, the stimulatory activity decreases. We have done other biochemical and cellular assays to assess effects on export. And these interfaces between the two proteins, including this one residue, are critical for regular mRNA export. So if we take this together, what we have done by capturing a few snapshots in the pathway combined with biochemical and genetic data is it allows us to place key factors. We separate this into two steps, the recognition and packaging of the mRNP by allele, TO, and UAP. This then allows the clamping of U56 onto the RNA together with the factor SRP to generate this novel intermediate where U56 is clamped on the RNA. The checkpoint has been passed. This mRNP presumably could freely diffuse in the nucleoplasm until it meets a nuclear pore complex and TREX2, where then U56 is unloaded and released such that the mRNP can pass into the pore aided by a so-called export factor. What is exciting about this model is that it would place the key factors conserved from yeast to humans and necessary for mRNA export into a single linear pathway. It would, at its center, place the ATPase, U56, and its RNA clamping cycle. and would suggest that in theory, a single round here can be achieved for a single round of ATP hydrolysis. Now, this model would also make predictions that are testable. For example, you would predict that if in a cellular system you interfere with some of UP56's interactions, you would have predictable outcomes. So for example, if you were to introduce a mutant of UP56 that can no longer bind to TREX2, but can still bind to the TOE complex, you would assume that this type of intermediate should accumulate. Similarly, if you were to mutate a non-overlapping interface of UP56 with the TOE complex, then no intermediate in the pathway should accumulate. This would allow us to test whether these are parallel or sequential steps in a pathway. And so these are comparative mass spec experiments comparing always wild type over mutant UP56 in IPs. And so as we would have expected, the TREX2 mutant leads to the accumulation of mRNA binding proteins here in red. If we prevent entry of UP56 into the pathway, mRNA components do not accumulate. This outlier is explainable by a technical artifact. And if we combine the mutations, there is no different, further consistent with the idea that these proteins likely act in a sequential pathway. What is really cool is that this model would now provide us with a general framework that can be expanded for regulation. You could imagine if you wanted to interfere or bypass steps of this pathway, you might want to do so by modulating the pink protein U56. And this is something that I will mention in just 2 minutes that we've explored very recently through this paralogous complex. So the story is this. I just showed you this work on the TREX2 complex. It had been known that there are paralogous complexes of this assembly. We saw the structures of them. The paralogs, one is called Lang8, replacing GAMP. The other one is called SAC3D1. Structurally, biochemically, they behave extremely similarly. All of them bind UAP. All of them can unclamp UAP from the RNA. Yet our cell biology data, and this is work we've done in collaboration with Torben Haig Jensen's lab, as well as Julius Brenneke's lab, suggests that the localization in cells of these assemblies is very different. SAC3D1 is purely cytoplasmic. Lang8 is nucleoplasmic. And GAMP, part of TREX, is anchored at the nuclear pore complex. So 3 assemblies, you do the same thing, yet in different cellular locations. We thought that the nuclear Lang8 complex would be particularly dangerous because if UP56 is a sensor of quality and export competence, having a free-floating nuclear complex that can compete with the bona fide export complex would be a huge problem. And so we wanted to understand what might be the function of especially this nucleoplasmic paralog of the TREX2 complex. And I will only show you 2 data points on this, but a big insight came essentially from the mass spectrometry data. This was carried out by Andri and Anna in Torben's lab. And so if you do an IP of the nuclear pore complex tethered TREX2 complex, you enrich for the nuclear pore. That's great. If you do, however, the IP of the nucleoplasmic Lang8 complex, its interactome looks rather different. And most striking is the enrichment of the nuclear RNA exosome, as well as 2 proteins that are called the PAX connection that link essentially to RNA decay. And so the hypothesis that emerged from this data is that perhaps these 2 assemblies act in competition with each other. One facilitating export of mRNAs and the other facilitating decay. To test this hypothesis in one of the many cellular assays, you would then predict that the RNAs that are sensitive to the perturbation of either TREX2 or Lang8 would overlap. And so this is shown here as a heat map. So these are after the Lang8 depletion RNAs that go up in the nucleus and the cytoplasm. And essentially similar types of RNAs respond in the opposite direction when you deplete GAMP, suggesting there is this competition between these 2 cellular assemblies. The core question that remains open is what would be the determinant for an mRNP to pass quality control and get out, or not pass quality control and get degraded. That is an area of active study. There's really interesting work from Yongcheng Shi's lab that suggests that there's a connection to delayed splicing. And that is something that we will explore in the future. So with this, I'd like to thank everyone in the lab for the really cool work. Ulrich and Max contributed a lot to the Lang8 and TREX2 work, and Belen and Matthias contributed to the earlier TOR-UAP work. We also have wonderful collaborators to thank, Torben and Julius Brennecke, as well as our very generous funding by Boehringer Ingelheim and ERC. And with that,